Hello friends, Ace Botane next episode. In this episode, we'll be proceeding with the principles of genetics, third one. So for convenience, I will share the screen. So it will be easy for you. I think screen came. So what all things we'll be learning in this episode? So left corner top, I mentioned the dihybrid cross, data on dihybrid, based on dihybrid cross, law, test cross, back cross, and their ratios, explanation to the concept of dominance, then as usual, question, theory, entrance. So first page you can find a representation from NCERT book, a typical dihybrid cross. So just to show it, I put the diagram. Now we'll be going to the cross. So first we should know what is dihybrid cross. Monohybrid you know. So what is dihybrid? In dihybrid cross, pea plants, cotton pea, Pisum sativum, Papilionaceae member, differing in two characters were taken. That is why NCERT there is a synonym given, inheritance of two genes. And what are the characters taken for this cross by Mendel? Round yellow seed. Diagram you can find the color, etc. Crossed with the wrinkled green seeds. Diagram again clear. And what is the genotype of round yellow seeds? Capital R, capital R, capital Y, capital Y. What about wrinkled seeds? Small r, small r, small y. And they represent parents. And from parents, when we analyze the gamete, capital R, capital R, capital Y will give only one type of gamete, that is capital RY. Small r, small r, small y will give only one type of gamete, that is small r, small y. And these two will be coupled, so to form F1. F1 will be capital R, capital Y, small y. Phenotypically, it is round yellow because one capital R and one capital Y is there. And the next cross, that is F1 will be usually self. That is a rule in genetics. Parents will be crossed and F1 onwards, it will be self. So capital R, capital Y, small y can produce four types of gametes. Capital RY, capital R, small r, capital Y, small r, small y. Same manner, the next one also. And to represent this four gametes into four gametes cross, we need 16 squares. And that is the diagram here given. And results of a dihybrid cross, where the two parents differed in two pairs, this one, two pairs of contrasting traits, seed color and seed shape. Here, below the diagram, NCERT statement. I think this page is clear. Now we can proceed to next page. Here I enlarged that checkerboard, and you can find how the gametes were written. Female gamete on this side, male gamete on this side. And here you can find it is the combination from this one and this one. That is how we have to represent. Here we can find from here and from here. That is how the combination proceeds. For this combination, this one and this one. I think that idea is clear. Now some data on this dihybrid cross, very predominant point for exam point of view. Phenotypic ratio of externally it appears it is 9:3:3:1. What about genotypic ratio? 1:2:1, 2:4:2, 1:2:1. And how we are getting this 9 genotypes? This is nothing but 1:2:1 into 1:2:1. Similarly, phenotypic ratio, we got 9:3:3:1. 3:1 into 3:1. I think that idea, it is clear. Now, total number of true breeds. True breeds means no heterozygosity, it will be 4. And you can find this line, it is true breed. Like capital R, capital R, capital Y, capital Y. Then capital R, small y, small y. Small r, capital Y, small r, small y, small y, where there is no heterozygosity. And total number of double hybrids, that is this diagram. Capital R, capital Y, small y. So two heterozygosity, double heterozygous. So this is that line. It's a very common question. We can anticipate. Now, total number of true parental genotypes. True parental genotype will be only two. That is one here. Capital R, capital Y, capital Y. One here. Small y, small y. Total number of parental phenotypes. It is 9 plus 1. One will be completely recessive. That is this one. And the 9, that is having at least one capital R and at least one capital Y. That is why I represented here. You can find capital R hyphen, capital Y hyphen. Hyphen stands for it can be filled with the capital R or small r. Same with the Y, capital Y or small y. There is no phenotypic change. So total will be 10. Now comes recombination frequency. For recombination frequency, there is a simple formula. Total number of recombinants by total number of progeny into 100. So it is total number of recombinants will be 6. Total offsprings or progeny will be 16. 6 by 16 into 100. And what are the 6 recombinants? That also I mentioned here. Here will be 2 boxes. Here only 1. Here also 2 boxes. So total 6 box representation will be there. What is meant by recombinants? Recombinants means new combination not seen in parent. That's all. New combination, recombination. And total 9 types of genotypes and 4 types of phenotype. So very direct data-based events for theory and runs based on typical Mendelian dihybrid cross direct NCERT, hoping that it is clear. Now, Mendel, he done monohybrid as well as dihybrid. Checked this monohybrid cross with the dihybrid cross. That means seed color, single character, yellow, green. Yellow with 12, green with 4. So ratio will be 3:1. Similarly, seed shape, round and wrinkled. Round will be 12, wrinkled will be 4. So again, ratio 12:4, 3:1. So very, again, a prominent question. Now, detail analysis. How many genotypes will be there in this checkerboard? So, pure round, pure yellow. This way we can take. Capital R, capital Y, only homozygous nature. So, we can find only one box. Hybrid round and pure yellow. Capital R, small r. So, 2 combination can be possible. Capital Y. So, 2 boxes will be there. Wrinkled pure yellow, that is small r. Then capital Y. Again, only 1 box will be there because no heterozygosity. Same with the pure round, hybrid yellow, capital R, Y, small y, 2. Hybrid round and hybrid yellow, maximum, capital R, Y, small y, 4. Wrinkled and hybrid yellow, again, small r, capital Y, 2. Pure round, green, capital R, small y, 1. Hybrid round, green, capital R, small y, it is 2. Wrinkled and green, all recessive, only 1. So total will be 4 plus 8 plus 4, 16. That is how 16 boxes came. Now, again, a simple representation, common, easy to follow. MH means monohybrid cross. Types of gamete will be 2. That is like capital T, small t, that way. Number of phenotypes, 2. Tall dwarf. Number of genotype, capital T, small t. So 3. Phenotypic ratio in monohybrid, 3:1. Genotypic ratio, 1:2:1. And when we come to dihybrid, almost double. That is type of gametes, 4. Number of phenotypes, 4. Number of genotype that become 9. Geometric progression. Phenotypic ratio 9:3:3:1. Phenotypic ratio 9:3:3:1. Genotypic ratio 1:2:1, 2:4:2, 1:2:1. So I think it is clear. Phenotypic ratio, genotypic ratio, monohybrid 3:1. Dihybrid 9:3:3:1. Genotypic ratio 1:2:1. Then dihybrid genotypic ratio 1:2:1, 2:4:2, 1:2:1. I think that is clear. No need to by heart all this data. Just practice it, check with the checkerboard. It will be easy for you. I think that idea is clear. Now we can proceed to law based on dihybrid cross. As you know, Mendel never proposed a law, some generalization of propositions. So here, generalization of Mendel. when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of the characters. There's a line given in NCERT book, page number 64, March 2020. Reddish 1-mark theory question for exam point of view, theory exam, define, or what is the law in connection with the Dihybrid cross. Name is law of independent assortment. Any character can couple with another character. That is the basic meaning. I think it is clear. Now we can proceed to next page. So 3 important crosses. NCERT wise only 2. One more is there. One is test cross, one is back cross, one is out cross. Each has got its own aim. First, what is test cross? Different definitions are there. Generally, when we say test cross, crossing tall in the sense F1 with recessive. And what is the aim in it? Aim is to find unknown genotype. That is the aim of test cross. In NCERT, there is a diagram for it. That is page number 59. Unfortunately, I haven't put it here. So page number 59, you can find a typical test cross that is illustrated. Now, what is a cross? Parents, capital T, crossed with a small t. Gametes will be capital T, small t. F1 will be capital T, small t. So parents will be cross. Now, when we come to test cross, capital T, small t crossed with the recessive parent that is small t. Everything represented here, you can find here. Very easy, simple. Gametes will be capital T, small t crossed with a small t. So phenotype and genotype will be capital T, small t. Phenotypic ratio will be 1:1, one tall, one dwarf. Genotypic ratio will be 1:1. So phenotypic ratio and genotypic ratio same in test cross. Famous question. In simple terms, test cross is the cross between unknown genotype to homozygous recessive parent. Aim is to find unknown genotype. I think test cross is clear. Now comes famous back cross. Crossing F1 hybrid with one of the 2 parents. Aim is to bring more parental characters, but sometimes dilutions may take place when the characters are formed. 2 possibilities are there. What are these 2 possibilities? Again, F1 will be capital T, small t. It can be crossed with the dominant parent, capital T. It can be crossed with the recessive parent, small t. So first one, this one, we can find combination will be capital T here, capital T, small t. So practically all will be tall. And second back cross is with the recessive parent. Again, F1 will be capital T, small t, cross with the small t, where we can find 2 combination, capital T, small t. So there we can find 50% tall, 50% dwarf. Now comes the third term. That is out cross. Crossing F1 with the dominant parent, aim is to overcome genetic diversity. Outcross. What is outcross? Outcross is crossing F1 with the dominant parent. Backcross also same item we observe here, almost same thing. And we can find it will be 100% tall. So these are the 3 crosses: test cross, back cross, out cross. And test cross was initially done by Mendel. I think that part is clear. Now we have to proceed with the test cross of dihybrid. Shown there. There also the value will be, ratio will be 1:1:1:1. Practically speaking, 1:1 into 1:1. 25% each. Now back cross of dihybrid 2-way possible that I written here. Dihybrid back cross, what will be the genotypes produced? Now a famous statement, all test crosses can be back crosses. Because back cross can be either test cross or crossing with the dominant. But all back crosses are not test crosses. Because back crosses can be crossing F1 with either capital T or with the small t. So 2-way possible, but test cross is always crossing F1 with the homozygous recessive. I think that statement is clear. It's an important one. So all test crosses can be back crosses, but all back crosses are not test cross. Now comes explanation to the concept of dominance. First, we should know what is dominance. Dominance classical definition is character which is expressed in F1 generation. When we represent alphabetic manner, we will be representing it as capital T and small t. And may think if capital T comes dominant, small t comes recessive. That is only alphabetic representation. Originally, what is the reason? It is not due to the value of the alphabet. It is concerned with the gene or genes. I think that idea is very clear. Now, a little bit detailed manner, because NCERT oriented, this idea, many people, for them, it will be slightly difficult, but it is comparatively easy. See, the genes contains information to express a particular trait, carrier. In diploid organism, it is 2n, there are 2 copies of each gene. We may call it as allele, allelic pair or pair of allele. These 2 alleles may not be always identical, like capital T, small t, as in heterozygotes, which modifies the information that a particular allele contains. Normally, normal allele will produce normal enzyme, and this enzyme normally converts substrate S to normal product. Theoretically, the modified allele could be responsible for the production of. This idea is very predominant for exam, NCERT direct. Either it will be producing normal or less efficient enzyme. Second can be a non-functional enzyme. Third is no enzyme at all. 3-way it can act, modification. So before getting into it, we should know what are enzymes. Enzymes are biological catalysts. You studied in biomolecules. While what are hormones? Hormones are chemical messengers closely linked with metabolism. So there is a connection, or we can say these enzymes and hormones are very predominant for metabolic events. Now in case 1, that is normal or less efficient enzyme, the modified allele is equivalent to unmodified allele. So no change. So it will be producing same phenotype or trait as in the case of silent mutation. Silent mutation means no change. But if the allele produces a non-functional enzyme, suppose there is no enzyme even, that is case 2 and 3, phenotypes will be affected. And how this effect comes? Unmodified, that is functioning, allele represent original phenotype, otherwise called dominant allele or wild allele. The modified allele is generally recessive allele or mutant allele. Now, what actually happens? Recessive trait is due to non-functional enzyme or no enzyme. That now we know. We can take typical example as tallness, a dominant feature. Plant height depends on amount of particular hormone. Hormone means metabolic regulating chemical. The quantity of hormone available, which is connected with the respective enzyme. That is why I told these two are linked. Suppose the enzyme is efficient, more hormones will be produced, resulting in dominant feature like tallness. If genes are altered, the enzyme become non-functional, or even no enzyme production, leads to less hormone and plant become even dwarf. This is how that originally works biochemically. I think that idea is clear. After that, as usual, 5 theory question and 5 NEET model question with the key. So what all things we learned in this episode that we can just rewind. So we started from dihybrid cross, then datas, law, test cross, back cross, ratio, explanation to concept of dominance. First page, we can find symbol representation of NCERT diagram. Now, second page, it will be expanded manner. What are the characters? What all will be crossed? And how many types of gametes will be produced from F1? And total representation in a typical checkerboard. Then the ratio. That is another important aspect, datas, genotypic, phenotypic, true breeds, double hybrids, then true parental genotype, parental phenotype, then recombination frequency. After that, what is meant by recombinants? Finally, at the bottom, we can find 9 types of genotypes and 4 types of phenotype. Then we proceed to checking by Mendel with the monohybrid and dihybrid cross, yellow-green, 3:1, round wrinkle, 3:1. And how these 16 squares will be formed, that is given here, another common question. Then here we can find monohybrid, dihybrid, number of gametes or types of gamete, phenotype, genotype, phenotypic, genotypic ratio. Then law based on dihybrid cross, definition given in NCERT book. Then test cross, back cross, out cross. What is test cross? What is back cross? What is out cross? Why it has been done? And what are the ratios? Then we entered into dihybrid test cross and back cross. Then all test crosses can be back crosses, but all back crosses are not test crosses. Then explanation of concept of dominance. What is meant by dominance? It is not the value of alphabet. It is concerned with the gene or genes. Then pair of allele, normal type, deviation type. Then it may connect with the production of normal or less sufficient enzyme, non-functional enzyme, no enzyme at all. In between, we explain, here you can find what is meant by enzymes and what is meant by hormones, how they function, is there in relation. Then how this dominant or recessive comes, connected with enzyme as well as more hormones. And that is how tall and dwarf will be produced. Then as usual questions, 5 theory question, 5 neat model question with a key. So this is the episode for today. And if you like it, subscribe all. Like it, share it, comment on it. We'll be meeting in next episode. So thank you. We can meet later. Take care.